How does cytogenetics work in leukemia testing? Imagine looking at the tiny building blocks inside your cells to understand what makes leukemia different from other diseases. Cytogenetics is a special way doctors examine the chromosomes inside leukemia cells to find clues about the disease. It all starts with collecting a sample of blood or bone marrow, which contains the leukemia cells. These samples are then sent to a lab where the cells are encouraged to grow and divide. When cells divide, their chromosomes become visible under a microscope, making it possible to analyze their structure and number. One common technique is called karyotyping. In this process, cells are treated so their chromosomes can be stained and viewed under a microscope. This allows scientists to see big changes like parts of chromosomes swapping places, known as translocations, or sections missing or duplicated. These large changes are often linked to specific types of leukemia. Karyotyping is considered the gold standard because it provides a clear picture of these major chromosomal alterations. Another powerful method is called fluorescence in situ hybridization, or FISH. This technique uses tiny fluorescent probes that stick to specific DNA sequences on the chromosomes. When viewed under a special light, these probes glow, revealing even small or hidden genetic changes that might be missed with karyotyping. FISH is especially useful for detecting specific gene rearrangements, like the Philadelphia chromosome, which is common in some types of leukemia, such as acute lymphoblastic leukemia. Polymerase chain reaction, or PCR, is another tool that amplifies tiny amounts of DNA or RNA from leukemia cells. This method is incredibly sensitive and can detect very small genetic mutations or changes. It is often used to find minimal residual disease, meaning small numbers of leukemia cells that remains after treatment. This helps doctors understand how well the treatment is working and whether the disease might come back. Next-generation sequencing, or NGS, takes a broader approach by reading large parts of the leukemia's genetic code. This technique can identify a wide range of mutations and chromosomal changes, even those too small to see with other methods. NGS provides a detailed genetic map of the leukemia, helping doctors tailor treatments to each patient's unique genetic profile. Chromosomal microarray analysis, or CMA, is less common at the start but can detect tiny duplications or deletions across the entire genome. These small changes can influence how aggressive the leukemia is or how it might respond to treatment. All these techniques work together to give a complete picture of the genetic makeup of leukemia cells. The information helps classify the leukemia into specific subtypes, which is crucial because different subtypes respond differently to treatments. For example, if the tests find the Philadelphia chromosome, doctors know to include targeted therapy with tyrosine kinase inhibitors. Cytogenetics also helps assess the risk level of the disease and guides decisions about how intense the treatment should be. In essence, cytogenetics combines traditional microscopic analysis with advanced molecular techniques to uncover the genetic secrets of leukemia. This detailed genetic information is essential for accurate diagnosis, predicting how the disease will behave, monitoring how well treatment is working, and planning personalized therapies. The process involves growing cells in the lab, analyzing chromosomes under a microscope, and using sophisticated molecular tools to interpret complex genetic data. It is a cornerstone of modern leukemia diagnosis and helps ensure patients receive the most appropriate care based on their unique genetic profile. Always remember to consult healthcare professionals for personalized advice and treatment options.